Thank you very much for the kind introduction. It's a real pleasure. Thank you, Jesus and the team for this nice invitation. It's truly a pleasure to talk here. I think this is my second talk in Valencia. I think my first one for my PhD thesis. So I'm really thrilled to be here. So I'm going to talk a little bit about the work that we have been doing since I moved to United States, to San Francisco, and in relation to abnormal patterns of neuronal activity. And particularly, we use mouse models of the disease because I think they can teach basic mechanisms of the disease. And so as you know, the brain is extremely complex. So my lab is interested in the pathophysiology of Alzheimer's disease. We use transgenic mouse models. Now we're using transgenic models, but also the knocking new generation of models to try to understand basic mechanisms of the disease. And the brain contains from molecules, synapses, neurons, circuits, and networks. And over the last 20 years has been a tremendous effort to understand what is happening at the synaptic level in Alzheimer's disease. And overall, there is an agreement that A-beta enhances synaptic depression and impairs short and long-term plasticity. However, when we and others start to look at the neuronal circuit level and also at the network level, we started to see that neurons become hyperactive. And even at the network levels, we observe hypersynchronization and even epileptic form activity. So this is a conundrum between what is happening at the molecular level and what is happening at the network level. And it's a little bit paradoxical. And in the lab, we are trying to understand this difficulty. But I really like this quote from Niels Bohr, that how wonderful that we have made a paradox. Now we have hope of making some progress. So the first evidence that we have that there was increased network hypersynchronization in models is when we started to do EEG recordings in this model, the J20 model of Alzheimer's disease, in collaboration with Jeff Nobles. And we observed that these mice, they develop hypersynchronized networks, and they have these silent seizures, no convulsive seizures. And this, you can see the non-transgenic and the APP mouse, it was not associated with convulsive activity. So that was very paradoxical observation in Alzheimer's disease, and a little bit perplexing because the general dogma was that there was a decrease of neuronal activity. Fortunately for my career, this has been reproduced through all the different models of Alzheimer's disease with familiar mutations that show a strong cognitive decline. So and I like also this quote, and I think it's particularly relevant for people studying neurodegeneration, and I think we have to embrace both sentences. This is a statistician, George Box, but it really fits in the current understanding of neurodegeneration. So it says that all models are wrong, but some are useful. And I think we have to be aware of the caveats of the model, but also we have to be persistent and try to understand what are the useful of the models and how we interpreted the results that we obtained from these mouse models, knowing the caveats that they have. So at the same time, there was a very influential paper from Columbia, Johns Hopkins, and Mass General that they show that the incidence of seizures in the Alzheimer population. And they have very interesting observation that the early onset was a very strong risk factor for the disease. And this probably was associated to familial forms that in this case didn't do the screening genotype. But in a recent review, we investigate all the different pedigrees associated with familiar Alzheimer's disease, and there are four causes. One is APP duplication, APP multiplication, presenilin 1 and presenilin 2. And around 40% of the pedigrees with familiar early onset, they do develop epileptic activity. And some of these, there are actually people that argue that some of these are syndromes of epilepsy, like Jeff Noble would do. So this is a recent study of neurology with a larger cohort of familiar, and I think this is the first paper that assess epileptic activity, specific epileptic activity. And this is a large cohort with 132 patients, and they observed that around 40% in a follow-up study, around 47% of the patients, they develop epilepsy and seizures. And this is the percentage of, according to the different mutations. And you can see that EPP duplication, 81% of the patients develop epilepsy, which is very, very high. And this is similar seen in Down syndrome that progress to epilepsy. So the most recent discovery, and this is by Andrew Cole and Mass General, he observed that is a very provocative. He actually did a study in which he implanted electrode in the foramen oval electrode, so they can record directly the electrical activity from the hippocampus. And he actually observed in a couple of patients of Alzheimer's disease that they develop seizure activity. So this is the regular scalp EEG recordings that you can not detect this epileptiform activity, and these are the intracranial foramen oval electrode. And they could see seizure activity in the two patients of Alzheimer's disease that was not associated with scalp EEG. So this highlights maybe the difficulty to detect epileptiform activity in this population of patients. So we could be underestimated the presence of this epileptic activity. So I like this quote, like, this is my quote, that maybe not all models are wrong and they can teach us something about the disease. So in the lab, we see the clinical seizure like a extreme manifestation of neighbor hypersynchrony that is associated with familiar Alzheimer's disease, particularly in pedigrees with very early onset and EPP duplication, but there is a gradient from clinical seizure epileptiform activity from network abnormalities. And we became very interested in network abnormalities, particularly in oscillation, to try to understand whether there was something beneath this clinical epileptiform activity. So one of the first evidence that we observe about a relation between oscillatory activity and the epileptic phenotype is when we started to do EEG recording. This is the raw EEG recording, and these are a spectrogram. So here it shows during 60 minutes the different power. So the color code is the intensity, the power, and this is the different frequencies. So the brain generates different frequencies that are associated with different behaviors and also are associated with different population of neurons and interneurons, coordinated neuronal activity. So it gives a very good representation of what happened, how active is the brain. So when we quantify the number of epileptiform spikes here, we see a very tight correlation between the increase of epileptiform spikes and reduction of gamma oscillatory activity. And here you can see the pattern. So there was very consistent across multiple mice, and this really shows us that this probably is related to interneuron dysfunction, because the gamma oscillations particularly generated by inhibitory cells. So this is the model that we have at the time. So we found that the power of gamma oscillation was reduced in the models of Alzheimer's disease, and this was tightly associated with epileptiform activity. And we thought that there was something wrong with the inhibitory cells, particularly with the 5-spiking inhibitory cells. So we show that indeed was the case, because we identify a voltage-gated sodium channel that is expressed in particularly inhibitory cells that is important for the generation of the action potential. And when we partially restore this level with a back transgenic mice, we restore some of the deficits in the gamma oscillatory activity, epileptiform activity, and behavior. So we think that this circuit is also important in human. So this is our mouse data. This is during active period, we see increase of gamma, and this is associated with the spikes. But in this setup, we don't know what is causal and what is a consequence. But however, a very nice paper for Matsumoto, they show that only during encoding, and this is important, only they were showing pictures that they have to remember, and to patients with epilepsy, and these are cases of epilepsy, not Alzheimer's disease. I wish some people from in the clinical setting of Alzheimer will do this experiment. So these are humans with epilepsy, only during the encoding increase the gamma oscillation, and this gamma oscillation is associated with the reduction of a spike, showing that it's a causal relationship that during encoding there is a change in oscillatory activity patterns that is actually beneficial in terms of controlling this neuronal hyperexcitability. So in the lab, we are now very interested to go deep into the circuits that are involved in the generation of gamma oscillatory activity. And as you know, the brain consists of pyramidal cells that are excitatory, pyramidal morphology, and then a set of neurons that are called interneurons that represent around 25% produce GABA, and they have diverse morphology. And in the lab, we're particularly interested to understand the emerging properties when these two cell types work together. And we are very interested in understanding timing, synchronization, and oscillatory rhythms, because these emerging properties of network activity are closely related to cognitive functions. And it has been that are affected in cognitive disorders, including schizophrenia, autism, epilepsy, and Alzheimer's disease. So we see the interneurons, they play key roles in many aspects. So they regulate of network function. They regulate critical states between the changes of states between active network and resting network. They regulate cognitive dependent brain network activity, oscillatory rhythms in terms of the high and low frequency oscillation, and they also regulate network excitability. And all these functions are important for memory functions. And in Alzheimer's disease, many of these aspects of network function are affected. For example, they have problems with brain state, sleep-wake cycle disruption, activity-dependent A-beta fluctuation day versus night. They have, during the initial states, they have, even in MCA patients and even in APOE4 carrier, there is a hippocampal hyperactivity, suggesting that there is an interneuron inhibitory dysfunction there, or also deficit deactivating the default network during encoding. So these are very early hallmarks of Alzheimer's disease. There is also EEG slowing, reduced gamma oscillation, and also we observe predominantly in the familiar cases that there is epileptic activity and seizures. So we see the interneurons as that provide, that can provide some answer regarding mechanisms of network dysfunction, but also some therapeutic entry points of intervention. So today I'm going to show you a little bit thing that we are doing in the lab. So we have a project that we are trying to understand the circuit alteration. So we are using optogenetics tool in different cell types to understand the circuit that produce gamma oscillatory activity, understand where is the problem there at the cellular level. So we have another project that I'll discuss a little bit more today about the using cell-based therapy of genetically modified interneurons, and we recently published that, and we are now working with Neurona Therapeutics to use human embryonic cells to try to see whether we can reproduce some of the data that we obtain with mouse cells. And then later I will talk a little bit about a project that we're very excited to enhance, to move some of these findings to the therapy. And we are trying to develop small molecules to enhance NAV 1.1 function to enhance the activity of inhibitory interneurons. So as you know, the interneurons are produced in the MGE, and they migrate into the cortex during normal development. And Arturo Arbaribulla, Scott Baraban, and John Rubenstein, a decade ago, they showed that these cells can be transplanted into adult mice, and then they perfectly survive and they colocalize and integrate well in the tissue. So we started a collaboration with Manuel Alvarez Dolado in the cabinet to use this technique, because he did a postdoc with Scott Baraban at UCSF, and we discussed the possibility to do this transplant in the models that have epileptiform activity. So by that time, we started to do an experiment in which we changed the molecular composition of the transplanted cells. So we use wild-type NAV1.1 overexpression cells, but also NAV1 deficient cells from a Dravet model, just to test how important are the interneurons and how important are the sodium the expression of the sodium channel. And then the host, we use the non-transgenic and the J20. So we produce two types of cells. One is fast-packing interneuron and probably parvalbumin cells, and then non-fast-packing interneuron somatostatin cells. And the key mechanism that we are producing in these cells with different levels of sodium channels is that we are changing the action potential kinetics. So these are recordings from fast-packing, slice recording after a year of transplantation from fast-packing interneuron and non-fast-packing interneuron. And you can see that the ones that are expressing that they have haploinsufficiency for NAV1.1, so they have reduction levels of NAV1.1, they cannot sustain firing rate. So the amplitude of the action potential diminishes. But interesting, when we express NAV1.1, we see that a shortening of the action potential. So this is the wild type, the NAV1.1 deficient cell, and this is the NAV1.1 overexpressing cell. And this was the case in the fast-packing and non-fast-packing. So this suggests that the expression of NAV1.1 is making the cells faster and accelerating also the excitability. This is some of the quantification that I just described to you. So interesting, when we transplanted these three types of cells in the mouse model, in the J20 mouse model, we observed that they have an interesting effect. So this is the open field. So the J20 mice, in general, the APP mice are very hyperactive. So we can test them before the transplant, and this is after the transplant. So we can observe that in red are the sham J20. So this is mice that receive the surgery but not the cell. And then we have in green the mice that receive wild-type cells, and then in blue the animals that receive the J20 that receive the NAV1.1. So we observe interesting, very mild or no effect of the wild-type cells, and then we see a very protective effect when we transplanted the interneurons with the NAV1.1. So this really suggests that the interneurons are important, but also the levels of NAV1.1 in the cells. So we observe the same thing when we study the fear condition and also in the Morris water maze, that the wild-type cells didn't give any benefit to the mice, but the NAV1.1 did. So we found a bunch of things that they were associated with a beneficial effect. That we see increased gamma power, reduction of epileptiform activity, and increased behavior-dependent induction of gamma oscillation. So we are actually very interested to translate this into the clinic, and we are developing small molecules that enhance NAV1.1. And for doing that, the first experiment that we want to do is to see how reproducible is this finding in another model of Alzheimer's disease. So by now we have been discussing the model of J20 mice, and this is another model. Is also an overexpression using different promotion, and in this case, express APP and presilinin. So these mice develop premature mortality that is associated with epileptic activity, and you can see a reduction of the survival. So these are, I think hundreds of mice in which we follow through months. So these are here in the J20. We have sex effects. The males have more premature mortality than the female, and the APP mice, they are equal. So when we test the NAV1.1 back, doesn't give any mortality, and then when we test with the when we cross these mice with the NAV1.1, we see a recovery of the mortality in the males and the females. So this move from the males from here to here and the females from here to here and here. So this really suggests that back transgene overexpression, and this is not targeted to NAV1 interneurons. This is back transgenic that is simulating pharmacological enhancement of NAV1.1 because it's using the endogenous regulatory promoter of NAV1.1. So wherever is expressed NAV1 will be expressed this back transgene. So this suggests that this is safe. So we were interested to see what happened later with the pathology of these mice. And in addition to the behavior that I showed you before, we also show that these mice, when they get older, they start to accumulate amyloid plaque in the hippocampus. So this is immunostaining for A-beta in the hippocampus. And we see that the enhancement of the NAV1.1 prevents the mortality, but also the presence of amyloid deposits in the brain. So we decided to go ahead with the project, and so we have 3 steps. The step 1 is to identify small molecules that enhance NAV1.1. The second step is the key improvement here is to understand whether these small molecules that we identify in HEX cells that are actually able to generate gamma oscillatory activity in brain slices. And the third step that we are starting now is to see what happens when we put this enhancer in vivo in mice. And so this is some of the results that we have. So we identify NAV1.1 enhancers in HEX cells. So we did a screening in a CRO with Evotec, and we screened around 50,000 compounds. We identify a bunch of these that enhance NAV1.1. And interesting, some of them, they are able to generate a gamma oscillatory activity in vitro. So for doing that, we are doing channel rhodopsin. So we are using channel rhodopsin to express in inhibitory cells, and we are able to generate gamma oscillatory activity in brain slices. For example, this is a slice in which we express channel rhodopsin in NAV1.1 interneurons, and this is the LFP recordings that we are seeing, and without stimulation. And after laser stimulation, we can see that there is an oscillatory activity we generate oscillatory activity that is associated, that is representing the gamma oscillation, because here we are activating specifically the inhibitory cells. So interesting, when we activate the interneurons here with the laser, we can see a very prominent induction of gamma oscillation. This is the 30 to 50 hertz power. And when we put an NAV1.1 enhancer, we see enhance from here. This is the baseline, enhance of NAV1.1 at the peak of the stimulation, but also at the baseline. And then this is strictly dependent of action potential, because when we block action potential with TTX, we completely block that. And interesting, and this is quite fascinating, this activity is really specific of gamma oscillatory activity. When we measure another frequencies, for example, the 6 to 12 hertz, we actually don't see enhancement during the optogenetic activation, and then we don't see changes with the NAV 1.1. So that means that we identify small molecules that they are active only when the interneurons are active, and they are able to promote gamma oscillatory activity without altering other oscillatory frequencies. So in the lab, we actually are very interested to understand what is the circuit that is important for the generation of the gamma oscillatory activity. And we were very surprised to see that because before the gamma generation has been attributed only to the parvalbumin cells. And these are seminal papers that they were published before. But all our manipulation that we do, so for example, when we do interneuron transplants of NAV 1.1 overexpression, or the slices that you just saw are the chanorodopsin is expressed in both types, in parvalbumin and somatostatin cells, we see a really powerful increase of gamma oscillatory activity. So Sandrine in the lab started to think to really understand what is the interaction between these two types of interneurons in the generation of gamma oscillatory activity. And so so she repeated the previous experiments. So this is in vivo recordings with silicon probes implanted expressing chanorodopsin only in the parvalbumin cells. And we, as previously showed, we saw that the increase of the intensity in the stimulation of these parvalbumin cells, it was associated with increase of gamma oscillatory activity. And we actually didn't observe that when we activated the somatostatin cell. However, when we activate both cell types, we see really a strong synergistic effect in terms of the generation of gamma oscillatory activity. So this, for us, is really quite fascinating. And this is the quantification to show the interaction between them. So the somatostatin cells, there is no increase of gamma oscillation. Parvalbumin, there is slight increase of gamma oscillation and much stronger when we activate both cells. So we went a little bit deeper to understand that and started to perform single unit recordings to identify the different cell types during gamma oscillatory activation. So this is the cycle of gamma oscillation that is 25 milliseconds, and this is the laser on that is 1 millisecond. So you can see the 5 spiking interneurons are extremely fast, so we turn the laser on 1 millisecond, and all they fire at the same time. And this is just one cell. And we have also identify non-fast spiking cells that they have an action potential that are a little bit delayed, and they have more tonic activity. So you can see during not only after the laser, but during the 25 millisecond cycle of the gamma oscillation. And this is the effect that we observe in the principal cells, that is a very strong inhibition. So I forgot to mention that if you're not familiar with these plots, that each dot here represents an action potential. So these are the action potential of the different cell types. So we became then very interested to understand to really why we get an enhancement of gamma oscillation. And actually, even Soltes show that there is a sequential action potential activity in between 5 spiking interneuron and somatostatin cells. So the 5 spiking interneuron fires first, and the somatostatin second. So the pyramidal cells receive first inhibition in the soma and then in the dendrite. And this happens across all these cycles, all different frequencies, including gamma oscillatory activity. So we think that the synergistic effect is associated with the timing of the stimulation that we are occurring. So we wonder how how functional is the gamma that we are generating, because the previous study have analyzed in anesthetized mice. So this is behaving mice. So we could measure the speed of the mice when they are moving, or the speed of the mice when we are recording. And we observed that when we activate the parvalbumin cells, we see a very nice synergistic effect with the speed. So when the mice are not moving, even though we stimulate, there is almost no induction of gamma oscillatory activity. So the induction of the gamma induced by the laser is when the mice are moving, suggesting that the gamma that we are generating actually enhancing the induction of one. So when we did the same experiment with the somatostatin cells, we actually observed the opposite. There was almost no induction of gamma oscillation, but when the mice are moving, actually the activation of the somatostatin cells impaired the generation of gamma. So this was very surprising and very interesting to them. And this talk about the timing. So if the timing of the activation is wrong at the cell type, there's an impairment in the gamma that is important for cognition. So when we activate these two, and this is still preliminarily, we observe that a really synergistic attempt that the mice don't need to move to have the gamma. So overall, this really suggests that different interneuron types synergistically cooperate to generate gamma oscillatory activity, and the sequence of the sequence of the action potential timing is very important. And so we did the same experiments with the U20 mice, and we observed, and maybe we can focus just on the top one, that this is the wild type, and this is the G20. We see that the G20 have a really severe impairment generating gamma oscillatory activity when we activate in the 5 spiking cells. And then, and we go, they're telling me that I have like 10 minutes, so I think I'm going to wrap up very quickly. And we went into what is happening. So I'm showing before LFP, that is, the overall network activity, but we have also the possibility to record individual cells. And here we have data showing the activity when we stimulate the 5 spiking cells. When we stimulate in the 5 spiking cells and record the 5 spiking cells, we see a really clear impairment in the firing rate of the inhibitory cells in this mouse model. And this is associated with loss of inhibitory zone in the perineal cells. For example, here we are stimulating parvalbumin cells and we are recording principal cells. So overall, the picture that is emerging from this cellular dissection is that we have the primary effect that we think is happening in these models is that there is a hypofunction of parvalbumin cells, and this is associated with a feedback forward inhibition to the somatostatin cells. And actually we see clear signs of enhanced activity and hyperactivity in somatostatin cells that is probably related to the impairment of the gamma oscillation. And I think this combination in these two cell types caused the reduction of gamma oscillatory activity that is associated with behavior. So as a conclusion, I think overall, inhibitory impairment interneuron contribute to network and cognitive alteration in mouse models of Alzheimer's disease, enhancing NaV1.1 interneuron function by genetic or interneuron transplant increases gamma oscillation, reduces network hypersynchronization, and improves cognitive functions in Alzheimer's disease. We identify small molecules that enhance NaV1.1 that promote interneuron-dependent gamma oscillatory activity, and interneuron cell types cooperate to generate gamma oscillation, and these mechanisms are affected in AD mice. And I think the overall conclusion, I think our data shows that enhancing interneuron function may restore oscillatory rhythm and cognition in neurological and psychiatric disorders linked to network hyperexcitability. So this is the people, some of the people that contributed to the lab and the funding, and particularly for the project, we collaborate closely with Manuel Álvarez Dolaro in the Cabimer, and I'll be happy to take any questions. Thank you.
Thanks, very nice and complex approach. Just a naive question. What happens with excitatory neurons? Because if there is hyperactivity, hypersynchrony, I may thought as an initial naive approach that is excessive excitatory.
Yeah.
Can be that an interplay with the regulation of the inhibitory? There is any
insights? Definitely. So we observe disinhibition. So in the excitatory cells, they show the standard hallmarks. So they have like reduced LTP, reduced excitatory transmission, reduced synapses, but at the same, and we observe at the same time disinhibition of the cells. So they are not synchronized. At least when we stimulate with the 40 Hertz gamma oscillation, we see a reduced inhibition of these cells, and the pattern of cyclic activity is not as tight as in the non-transgenic. So it's a both, when both cell types are affected, and it's the way, the cooperation in the both, that is the thing that is more affected. Because the small changes in interneuron and the small changes in pyramidal cells, when you put in a scenario of a network function, they are both exacerbated.
I guess that this Dr. Shai at MIT,
yes,
the one that has a project called Genus.
Yes.
And this project is, in this project, they try to induce gamma oscillations for to prevent or to cure Alzheimer's disease. Can you comment the possible relation between your project and Dr. Shai project?
Yeah, it's actually very related, because she showed that she showed first enhancing gamma oscillation by sensory stimulation can reduce pathology and also neurodegeneration in some tau models. And I think it's very tightly related with our models. So we are using the interneurons to drive gamma oscillation, and we are using pharmacology to enhance this rhythm. And Li Wei Sai is using sensory stimulation, auditory stimulation, and then she observed similar effects in terms of cognition. The only thing that it has to be determined in the sensory stimulation whether if the interneurons are driving the effects or not, because they have not done like single unit recordings during sensory stimulation and isolated specifically the interneurons. So I was wondering whether they are driving more the pyramidal cells with the sensory stimulation, the layer 4, the thalamic input, and this drive the interneurons after. But the circuit mechanism of their stimulation is not completely clear to me, but it's complementary with our approach. Thank you.
Very beautiful, Jorge. I wonder if you think on a model, in a model where you have a clean model to perturb gamma oscillations, doing directly and targeting directly the PV neurons. So what do you expect what's going to happen in terms of, let's say, something like dementia or Alzheimer-like phenotype, just going directly in a clean model to the gamma oscillations? What would you expect?
I think will be beneficial. So targeting, what do you mean by targeting?
What do you mean in diminishing?
I think there would be.
So thinking about the causality.
I think all the data that we have suggests that even without any manipulation, that when gamma oscillation goes down, we have hyperexcitability in the cortex that probably is associated with the reduction of firing rate in the paroalbumin cells. I think reducing paroalbumin cell activity will be detrimental not only for Alzheimer's but also for controls.
I have a second question regarding the homeostatic plasticity. You know that all these changes in homeostatic plasticity will establish that you need to maintain the firing rate of a circuit. Yeah. So I wonder, in your models, have you thought about that, or how do you think this thing could be?
Yeah, we think, yeah. So for example, we don't know. That's an interesting question. We think that the problem, there is like two parts of the problem. One is the excitability. So there is intrinsic excitability that is reduced by the NaV1.1. And this could be, it may be a compensation of the, epileptic activity that they have, these mice. So we have not, unfortunately, in our mouse model, both, epileptic activity and inhibitory dysfunction are associated. We have not been able to eliminate one and not the other. So probably, the internal regulation is the homeostatic regulation of the firing rates of paroalbumin cells are definitely very important, but we have not been able to manipulate them specifically.
Yeah, I would like to also ask something. Has the diversity in gamma oscillations in the human population been studied?
Yes.
Could one propose that we have is a predisposing factor, for example?
Yeah. So there is very clear evidence of particularly the coupling of gamma and theta. So gamma and theta is very, very diminished even in MCI. And we actually observe the same phenomenon in the models of Alzheimer's. So the power is difficult to study because the total power is difficult. We change a little bit with the methodology, but the coupling between gamma and theta, the phase amplitude coupling between theta and gamma is a very good biomarker for MCI and Alzheimer's disease.
It's easier to gather that kind of data than using EMIT analysis or biomarkers in blood, right?
I think we are proposing that in the project, in the clinical project, we're proposing that as a biomarker because we also see a restoration with the NaV1.1 instead, and it's relatively easy to evaluate with EEG recordings from human.
Any more questions? No? So thank you very much, Jorge, for this excellent talk and for coming.